Hi, everyone. I'm Sreenivas, a third-year combined bachelor's-master's student in neuroscience. Today, I'll be discussing my research into the post-transcriptional mechanisms of neuropathic pain at the Meffert Lab. Neuropathic pain is a major medical problem caused by a lesion or disease of the somatosensory nervous system. The most common outcomes of neuropathic pain are hyperalgesia and allodynia. Hyperalgesia is an increased sensitivity of pain under the same stimulus intensity. This means that responses to normally painful stimuli are pathologically amplified. Allodynia is another format of hypersensitivity, which involves previously innocuous stimuli now becoming painful, or there is evoked pain in the absence of any stimulus. This diagram shows how neuropathic pain is developed following nerve injury. After injury, the injured primary sensory neurons will have altered sensitivity to mechanical, thermal, and chemical stimuli. This will cause ectopic action potential generation throughout these nerves and finally lead to altered synaptic transmission. However, the uninjured primary sensory neurons also show this kind of ectopic action potential generation. This ectopic transduction is mediated by many changes in both the peripheral and central nervous systems. These changes can be triggered by neurotrophic factors and other chemicals released by surrounding Schwann cells and immune cells following nerve injury. Before discussing the connection between the Lynch-1,8-Let7 pathway and neuropathic pain, let's first characterize the Lynch-1,8-Let7 pathway. Let7 or lethal 7 is a microRNA, which recognizes complementary bases in the 3' re-TR of the mRNA of many pro-growth genes and causes their degradation. Lynch-28 is an RNA binding protein with 2 RNA binding domains, including cold shock domain and zinc finger motif. Lynch-28 will recognize pre-Let7 transcripts and recruit a TUT4 protein to degrade the Let7 transcripts. This, in turn, causes the de-repression of pro-growth genes. Work in our lab has found that the rapid turnover of Lynch-28 protein is blocked by the binding of TRBP. TRBP binding to Lynch-28A is stabilized by MAPK activation. We also found that MAPK activation in the absence of neurotrophic stimulation still causes the stabilization of Lynch-28 and the downregulation of Let7. One of the first experiments we did in our lab was to test whether nerve injury can induce any changes in the Let7 microRNA family. We found that for each Let7 microRNA, there is a decrease following nerve injury at day 5 on the ipsilateral side. The control MIR132 did not have any significant differences. So putting all of this data together, we want to understand what roles do Lynch-28 and Let7 play in neuropathic pain. In order to study the mechanism of neuropathic pain, we use 2 different models, the SNT and SNI models. The SNT, or sciatic nerve transection model, uses the entire transection of the sciatic nerve and the peroneal, tibial, and sural nerves within it in order for us to compare the expression of genes like Lynch-28 and the levels of Let7 between the ipsilateral and contralateral sides of the injury. The spared nerve injury, or SNI model, allows us to compare the expression of Lynch-28 and other proteins between injured fibers from the tibial and peroneal nerve and uninjured fibers from the sural nerve on the same side, the ipsilateral side to the injury. Following SNI or SNT, we use the von Frey assay to test for pain sensitivity in mice. In the von Frey assay, we use von Frey filaments of increasing diameter to transduce increasing forces to the hind paw of these rodents. Previous data from the von Frey assay in the Katarina lab from collaborator Sangwan Jian has shown that there is a significant difference in the pain sensitivity of Lynch-28 conditional knockout mice and control mice. These differences become significant at and around day 7 following nerve injury. In summary, the loss of Lynch-28 in sensory neurons inhibits the development of hypersensitivity to mechanical stimuli, and LET7 growth suppressor microRNAs are downregulated following nerve injury. So if Lin28 is required for the full development of neuropathic pain, is there a change in Lin28 mRNA or protein following nerve injury? And how is this change spatially temporally distributed? In order to analyze the spatial temporal distribution, we used RNAscope. RNAscope is a single molecular detection fluorescence in situ hybridization method that allows us to detect individual mRNAs. And by using RNAscope, graduate student Shin-Bei Lee in our lab did a time course to test Lin28 mRNA expression at days 3, 7, and 28 post-SNI and dorsal root ganglia. Shown are the Lin28 mRNA distribution at 3 days post-SNI. ATF3 is a transcription factor which is highly expressed in injured cells. NUN identifies neurons, and the hex stains identify nuclei. The injured cells on the ipsilateral side at day 3 had a higher level of Lin28 mRNAs than did uninjured cells on the same side. All the images shown here are maximum intensity projections of Z-stack images. Shin-Bei and I co-authored a protocol published in STAR Protocols for the analysis of mRNA expression in DRG and sciatic nerve using RNAscope. Much of my contribution to this protocol involved the development of a computational pipeline for the spot counting of RNAscope images. In order to quantify the total Lin28 mRNA puncta number in DRG cells, we used FishQuant, a MATLAB toolbox. The first step of this quantification involves the drawing of cell and nucleus outlines using the NUN and hex channels respectively. Secondly, discrete puncta are analyzed using the local maximum spot detection method. The parameters for detection are manipulated by assessing the program calling of puncta boundaries, followed by visualization. Lastly, the compound puncta or clusters of puncta are quantified using a point spread function. An average punctum is identified using the average fluorescence intensity and volume of all the discrete puncta, and a 3D point spread function based on this average punctum is then fit to all of the clusters. And the resulting peaks are called. The number of peaks is taken to mean the number of present puncta within the cluster. I also developed an ImageJ macro to automatically analyze the mean, maximum, and minimum gray values of GFP fluorescence in cells outlined using FishQuant. Though this did not directly produce experimental data, it allows us to quantitatively assess the infection efficiency of our AAV constructs that co-express GFP. Another script I developed provides a function not available in FishQuant. FishQuant does not have a way to separate cells from a single outline file, but since we want to be able to quantify differences in Lin28 expression between ATF3 positive and ATF3 negative cells in the same image, that was a function that had to be provided via an ImageJ macro. This macro shows the user a maximum intensity projection of the ATF3 channel, and the user can then select which cells in the image are ATF3 positive. The macro then automatically creates 2 outline files, one containing only ATF3 positive cells and one containing only ATF3 negative cells. Shin-Bei also performed RNAscope for Lin28 mRNA expression in a time course post-SNT surgery in sciatic nerve tissue. MAP2 identifies neuronal processes, and S100 stains for surrounding Schwann cells. All the images shown here are orthogonal projections of 3D Z-stack images. Our method for analysis of sciatic nerve tissue differs from the method for analysis of DRG tissue. The reason is because FishQuant is limited in its outlining in that a 2D outline is extended across the 3D Z-stack cylindrically. However, since Schwann cells envelop the neuronal processes in 3D, a 2D outline could not be extended across all Z-stacks without intersecting both Schwann cells and neural processes. This is a problem. So we used Imaris to conduct the quantification. The first step in Imaris quantification is to create virtual masking surfaces for all the channels. The first of the surfaces is a set of spots for the Lin28 spots in the image. We measured the XYZ diameter and intensity thresholds of a few Lin28 puncta and fed those parameters into the Imaris spot identification tool, which then identified all Lin28 puncta in our images. The second step in the quantification was to generate virtual surfaces for both the MAP2 and S100. During the surface generation, we adjusted the threshold of the mask to make sure it covered all the staining perfectly. The last step is to create filters for the virtual spots to identify which spots of Lin28 are co-localized to MAP2 and which one of them are co-localized to S100. Since Imaris doesn't have native support for this automatic assignment, I developed an Imaris extension to assign puncta to the MAP2 surface and to the S100 surface. However, since there was some unavoidable overlap between the MAP2 and S100 surfaces, some puncta seemingly co-localized to both surfaces. To deal with these redundant puncta, we decided to remove them from the S100 channel. The data I most recently analyzed characterized the spatiotemporal distribution of Lin28 mRNA within the injured sciatic nerve of SNI mice. At around day 3, we noticed that there were some significant differences in Lin28A and B expression in both neurons, indicated by MAP2, and glia, indicated by S100. This was particularly of note in the spared sural nerve. This is quite interesting and suggests that Lin28 expression within uninjured and injured fibers are temporally distinct, at least at day 3. The differences in Lin28 mRNA count are particularly large at around day 7 post-injury for the Lin28 homologs Lin28A and Lin28B. Lin28A is significantly upregulated within neurons and glia of the injured stump, and Lin28B is significantly upregulated in the glia of the injured stump. At around day 28 post-injury, levels of Lin28 can still be significantly elevated, but are reduced in comparison with levels at day 7. The Lin28 homologs are significantly elevated in the processes of some injured neurons and associated glia. In conclusion, we've observed the upregulation of Lin28 homologs in the injured distal processes at around day 7 post-SNI, where the differences in pain sensitivity between Lin28 knockouts and wild-type mice become pretty significant. And this is particularly significant in the injured nerve stump. We have also found that there's a temporal distinction between Lin28 upregulation in injured and uninjured fibers, at least at day 3. And we've also observed, not shown in this presentation, aggregates of Lin28 in the distal nerve stump. This is a topic of ongoing investigation in our lab, as we're unsure whether they're actually localized to neurons. Some experiments we've conducted very recently make us suspect that they are localized to mononuclear immune cells, but we are unsure of the specific cell type at this time. As for future experimentation and directions, I hope to investigate the role of Lin28 in hyperalgesia following nerve injury by determining whether Lin28 gain of function on its own is enough to trigger hyperalgesia. We'll be using a Lin28 AAV flex construct to inducibly trigger the recombination and overexpression of Lin28 in sensory neurons. Thank you so much for watching my presentation. I'd very much like to thank my mentor, Dr. Molly Meffert, and the collaborators, Junbei Li and Daniel Shen. Bye-bye.